So my talk today is titled a little ambitiously, I would say, because you'll realize at the end of the talk, it says Bye-bye Biopsy, Extracting Muscle Tissue Composition and Microstructure from MRI, Initial Validation to Biopsy and Application to the Aging Muscle. We'll say we are not quite there as yet to say bye-bye to biopsy, but this is the first. Doesn't it go next? So let me give a little background of the diffusion process that we are measuring by MRI. And so what we mean by diffusion tensor imaging or diffusion imaging. So diffusion is something that all of you are familiar with. A water drop in a beaker of a colored water drop in a beaker of water. You'll soon see that that color has diffused. And basically diffusion is has a formal definition of random displacements and is triggered or driven by internal thermal energy. So the higher the temperature, the faster the diffusion. So if you were to observe this diffusion or if you were able to track a particle as a function of time, then you'll find that these molecules, say water molecules, for instance, diffuse further away from the starting point. And this mobility is characterized by a physical constant, and that name is given the name diffusion coefficient. Sometimes you'll find that referred to as apparent diffusion coefficient, ADC, but they all kind of refer to the same thing. How fast can a water molecule, in our case, it's mostly only we are looking at water molecules, how fast water molecules diffuse in a particular structure. So if you consider large structure like, or if you consider like a beaker of water, and if you were to track its motion, then it would be the same in all directions. That's what is called isotropic. But in small structures, and essentially initially all this was started by looking in the brain, but now our work is all on the muscle. But axons and muscle fibers have structure. So this is kind of to show, this was more an axon image, but it gives you the idea that if the red is the is the random displacement of water molecules, and you can see it's random, so it takes any direction it wants. It'll translate to different locations, and if it's within this medium here, then it's not restricted. It may be restricted by some other molecules, but is not restricted by what is shown here, the membrane over here. So you can see that there is restricted diffusion if you're looking at small structures, and by small I mean small compared to the diffusion lengths or the area, or the distance over which the molecule can diffuse in the time of observation. And so you can see that if you have a long fibrous structure like this, there's less restriction for motion along the fiber, and there's more across the fiber. So you can kind of see there's a factor of 3 difference in the mobility coefficient. So that's what is exploited in diffusion tensor imaging in order to get you would have seen some cool pictures maybe of white matter tracks in the brain. A similar thing can be done for to track muscle fibers, to exactly know how the muscle fibers are going. So I'll show the sequences, briefly describe the sequences, but from the data that we acquire using diffusion gradients in different directions, we can actually get the mobility in different directions. So basically, a mathematical operation is done in order to get the diffusion coefficients in 3 perpendicular directions, kind of visualized as a schematic as an ellipsoid. So the longest axis of or the longest diffusion, the largest diffusion mobility is along the muscle fiber. So if you knew its value and if you knew its direction, as is shown on the little right insert here, you could actually track the fibers. And then the other 2 diffusions can give you some idea about the structure of the fiber itself. So very briefly, I'll be showing results from 2 different types of techniques used in the MRI to measure these diffusion mobility coefficients and anisotropy. And the first one is just maybe it's available on all the scanners. It's called spin echo diffusion imaging. And it's a very fast technique because if you're sensitizing the image to very small motions, You have to make it fast enough so that it's not sensitive to larger or gross motions or physiological motions. So this is like a 100 millisecond scan that way. And it's achieved by putting these two green, what we call diffusion gradients, magnetic field gradients. And you can see that if the if you have a gradient in one location as shown here, another gradient at another location, if it was completely static, the effects of the first gradient undoes whatever the second gradient will undo all the effects of the first gradient. But if it moved to a different location, it'll get a different magnetic field and the signal will be reduced. This reduction in signal is what we are measuring. And the same, there's another way, but you can see that it's quickly decaying over here. That means you don't have much time to let the spins or the water molecules diffuse, and that's this diffusion time. So there's yet another sequence where you can preserve the diffusion and let it evolve for a longer time before you make the measurement. All I can say is compared to this diffusion time, we can change the diffusion time for over much longer lengths, including up to 500 milliseconds, and still be able to measure the diffusion. Idea behind is that the longer we observe, the more we are going to be able to detect as these particles interact with the membrane. So that means you will be able to see differences between different size fibers, like for instance, with atrophy, you may have a smaller fiber, so maybe the effects are larger if you see the diffusion mobility after you observe it for a longer period of time. So we are using both these sequences. Just to show our initial work before this modeling, even the plain diffusion can give you a lot of information. For instance, I've shown here the tracts just colored differently to show you how the fibers run from distal to proximal in the medial gastrocs. And from that, we were able to get both the fiber lengths and the pennation angles. And we saw, as seen before from ultrasound studies or biopsy studies, the length decreased with age and the pennation angle also decreased with age. And this color map here, color coded map, so to say, is giving you an idea of the direction of the fibers, which was used, in fact, in tracking these actual fibers. So these are not lines drawn manually. These are actually tracked using the vector direction of the fibers. And when we looked at the diffusion eigenvalues, you found that there were small and subtle changes, and essentially not changes exactly like we anticipated, because we thought the fibers will atrophy and the third diffusion value actually gives you should be much reduced, but we found them all increased. And then the idea is that there could be two different mechanisms. That means an increase in the extracellular volume accompanied with a decrease in fiber diameter that might preclude us from making any judgment on which particular parameter is changing, because we are only making gross measurements. That led us to the modeling project. So the introduction is coming only now. So what we really, to summarize, we felt that the gross measurements were not giving us enough, were not able to let us make enough inferences about the microstructure, and one needs to go to a model that links the microstructure to the gross measurements, because we are not looking at any measurements at the scale of the fiber diameter. Our voxels are more like 2.5 millimeters, especially in diffusion. So we need a model to link these two, and that's what we tried to do for using two models. One is aging, the other is disuse atrophy. And our results earlier showed that the loss of muscle force only could be explained in part by the loss of muscle mass. And so we wanted to see what other microstructural parameters are changing that potentially could clue us into how the diffusion values were changing, the gross measures, and how that was related to muscle force loss. So one of them could be that the extracellular matrix is remodeling and maybe the collagen content. So that was the objective, to monitor the changes in diffusion tensor indices with aging and disuse, and to relate these macroscopic quantities to the underlying tissue structure. We use two models. One is a two compartmental model for the disuse and aging, and another, a simpler model with fewer parameters to fit, which is called a random permeable barrier model that fit to the time dependent diffusion. So I showed you the second sequence was able to give diffusion as a function of time. So it fitted to the time dependent diffusion. So the methods are basically, we had 7 subjects for the ULLS pre and post suspension. For the aging study, for the two compartmental model, we had these 9 young female and 8 senior. And then for the RPBM model, that is the random permeable model, we had 7 young and 8 senior. And in addition, many of the subjects also had biopsy of the muscle tissue. So we had biopsy samples for the model 2 correlation. So I won't go into this model itself, but all I want to say is that the model is complex, and therefore the fit is very prone to noise and fitting accuracy, which is very important in model fitting. So the first model essentially is two compartmental. That means it proposes the diffusion as the total diffusion that we measure as the diffusion in the intracellular region and the extracellular region. And the parameters, in addition, this parameter had something that was interesting to us, the fiber ellipticity. So is the fiber in cross section circular or is it elliptical, meaning um, it has an elliptical cross section. And the other parameter fit was mean fiber diameter, intracellular volume. Intra plus extracellular volume is 1. So this is the intracellular volume fraction. It also modeled the collagen content in the endomysium and the intracellular residence time. That means the average time that the water molecule resided within the cell. So it's actually a powerful model, but the only caveat in that is when you try to fit a single measurement to 5 unknowns, then you're asking for a little bit for trouble. So that was the first model. The second model is easier, and basically it was, as you can see, the model was randomly placed membranes. So it's like if you take a cross section of a muscle tissue in biopsy, you kind of just see some variations in the fibers, fiber diameters, and you're able to, and so that was how it was modeled. So it doesn't explicitly model the diffusion in the endomysium or anything related to the endomysium. It's just membrane permeability is modeled. So the was a simpler model and was only using 4 variables. So basically we fitted to the permeability, the effective volume fraction, and D naught. So let me go fast. I think this is, so I'll just go to directly to the model. So the two compartment model shows the ellipticity actually decreases, meaning the greater the ellipticity means anisotropic the fiber in cross section is lower from 1. So 0.5 means 0.55 means is very elliptical, and post it became 0.95. Same with young and old, the ellipticity, uh, the fiber cross section became more circular. And we didn't find as much difference in the others. In fact, for the fiber diameter between young and old, we didn't see much of a difference. Though we saw some difference between the pre and post for the disuse atrophy. So it shows that we can get some parameters out that could be related to the real microstructural changes. And intracellular volume actually decreased here a little bit and didn't change much. So in the aging, our fitting was not as good as maybe what the disuse atrophy fitting yielded. Basically, it said that the fiber cross section became more circular and that there was a decrease in the fiber diameter, both of which is as anticipated. The ellipticity is probably not anticipated, but when we look at our strain measurements that Shantanu just talked about, we find that the strain is very anisotropic. So that means deformation only occurs along one direction and not in the other. And that may actually be related to the disuse atrophy, that when you do not have a loading on the muscle and you don't have the deformation that is stretching only in one direction, the fiber cross section may tend to become circular. because there is no load on it, stretching it preferentially in one direction.
5 minutes included the discussion.
Okay, so then this is the results from the STEAM DTI, which was the diffusion, time-dependent diffusion. And again, I can summarize all these results. You can see the fit is actually good. So the red points are the experimental data and then the dotted line is the fit. So you can see a very good fit to the model. But we did not see the anticipated results in myofiber diameter. We would have thought it would decrease, marginally increase, not significantly though. The only thing that really decreased was the volume fraction. So that had some, that has some implications. The biopsy results, I'll just say that the only thing that the two important findings from biopsy of the same subjects, by the way, was that fiber CSA tended to decrease in old subject, and that the main thing they found was that the collagen matrix is the orientation changes from 20 for fibers in young to 30 degrees. So we tried to find some correlations between the model-derived diffusion parameters and the biopsy parameters. And there we found that collagen 6 had the maximum number of correlations. It's too premature to conclude anything, but collagen 6 is the one collagen that was densest around the individual muscle fibers. So it's possible that it is actually modulating the diffusion, meaning how much the mobility of the water protons is. So we think there is a chance that this could lead to some microstructural parameters that we can actually derive from the diffusion that may reflect what the biopsy is actually measuring. So that's our ultimate goal. I actually explained some of the findings of the two compartmental models. So I'll just go to the discussion of the RPBM model, the second model. It did not show the anticipated in decrease, but I have to say that the biopsy did not actually give us the ferret diameter. So there is a chance that the biopsy measurements of the diameter may be influenced by the angle of the fiber cut. But there was a positive correlation between the mean residence time, that is the mean time that the water molecule resides within the cell, between the muscle fiber and the fiber CSA as measured by biopsy. So that's a good correlation that if the size is larger, then it's going to reside for longer because it's going to take longer to diffuse out. And as I indicated, collagen 6, which is densely around the individual fibers, seem to have the most correlations. Membrane permeability was not measured in the biopsy, but if it's measured, then we have we are able to get that from the modeling. So that could be a direct correlate as well. So in conclusion, we have to say that though these are preliminary results with modeling, and modeling is subject to a lot of constraints because of these parameters may be very sensitive to the noise in the images, it still shows potential. We're excited by the ellipticity findings from the two compartmental models, and also the some of the correlations with collagen 6 that might indicate that collagen in the endomysium also modulates the mobility of water molecules. And from that, we could make inferences from diffusion alone, which might then, in the ultimate, in our ultimate goal is that we get rid of biopsy and substitute it with non-invasive imaging techniques. Thank you.
Marco?
Thank you very much, Usha. This was a very interesting presentation. And one remark, obviously time was against you, but this technique combined with the strain tensor also enables us to measure muscle deformation upon contraction, isn't it? And is
yes, that's very yeah, that's a very good observation. And in fact, we have been, Shantanu did not talk about it, but we also have DTI and the opportunity to do fiber aligned strains. So the strains along the fiber, so that could give us more physiological parameters about the deformations. I mean,
Yeah,
yeah,
and we hope
that's-
to use this technique for estimate the lateral force transmission.
yes, we could. I mean, in the sense that, yeah, if you knew the fibers and then we were able to get the strain in the fiber cross section, because we do get the strain in the fiber cross section, but as he indicated, there's an angle between the maximum values of these strains and the fiber. So to be able to align them exactly along the fiber, we'll be able to get parameters of, say, radial expansion, and these could I mean, we are saying, Marco, is that the shear strain that we measure from the strain measurements is related to the shear in the endomysium, though we don't have a way of actually through the measurements pinpointing where the shear is coming from, because a voxel contains hundreds of fibers and connective tissue.